Hello everyone, this is chapter 3. There will be 2 lectures for this chapter. We will begin to discuss acid-base chemistry and we'll look at its role in organic reactions and mechanisms. This chapter lays the foundation for many of the forthcoming chapters, so let's get started. There will be 2 videos for chapter 3.1. The first covers acid-base theories and the second looks at the strengths of organic acids and bases. There are 3 main acid-base theories, and I've included the faces of each of the people that were involved with their development. The Arrhenius acid-base concept, developed by Svante Arrhenius in 1880, classifies a substance as an acid if it produces hydrogen ions or hydronium ions in water. A substance is classified as a base if it produces hydroxide ions in water. This way of defining acids and bases works well for aqueous solutions, but acid and base properties are observed in other settings. Other ways of classifying substances as acids or bases are the Bronsted-Lowry concept and the Lewis concept. The Bronsted-Lowry theory, after Johannes Bronsted and Thomas Lowry of 1923, classifies a substance as an acid if it acts as a proton donor and as a base if it acts as a proton acceptor. This works both in aqueous and in non-aqueous solution. This theory was needed to be developed because Arrhenius' theory had a very narrow viewpoint of what an acid or a base is. The Arrhenius theory was hard to accept at the time because knowledge on atomic structures and distinctions between hydrogen ions and protons were not as clear as they are today. The Arrhenius theory did not include the behaviour of dry acid-base reactions, i.e. non-aqueous ones. Now, the Lewis theory, after Gilbert Lewis, also of 1923, can be applied to systems with or without solvents. In the Lewis theory of acid-base reactions, bases donate pairs of electrons and acids accept pairs of electrons. Seeing as the Arrhenius definition is narrow to aqueous solutions, we will not consider this acid-base theory any further. So let's take a look at some specific examples of the Bronsted-Lowry acids and bases first of all. In the Bronsted model, water is the active species in this reaction. It accepts a proton or a hydrogen ion and forms a covalent bond, hence it behaves as a proton acceptor. Therefore, the hydrogen atom of hydrochloric acid behaves as a proton donor. When this happens, a conjugate acid-base pair is formed. These can be interconverted back into the reactants by the transfer of a proton. The Bronsted-Lowry base, water in this case, gains a proton from hydrochloric acid. Therefore, the conjugate acid of this base, water, is the hydronium ion, whereas the Bronsted-Lowry acid, hydrochloric acid, donates its proton to water and forms a chloride ion as the conjugate base. Now, Lewis had a different model for this behaviour, which includes the use of arrows for the first time. In the Lewis model, the hydrogen ion is the active species. It accepts a pair of electrons from water to form a covalent bond. This is the dative covalent bond that is highlighted. Lewis bases donate pairs of electrons, and Lewis acids accept pairs of electrons. We'll revisit this reaction later on. This is an example of an aqueous reaction that occurs in water. But what about the reactions that don't occur under aqueous conditions, but can still behave as acids and bases under the Lewis concept? In this reaction, there are no protons being transferred. Rather, the electrons are being transferred, and these form bonds, which is similar to the previous example. The Lewis acid-base theory explains why borane, BH3, reacts with dimethyl sulfide, on the left in blue. Borane, a trigonal planar molecule, reacts because the electrons can only be found in 3 places in the valence shell of the boron atom. As a result, the boron atom is sp2 hybridized, which leaves an empty 2pz orbital on the boron atom. Borane can therefore act as an electron pair acceptor or as a Lewis acid, which we called an electrophile, i.e. electron-loving. It can use the empty 2pz orbital to pick up that pair of non-bonding electrons from our Lewis base, dimethyl sulfide, which is also known as the nucleophile, i.e. nucleus-loving, to form a covalent bond. Borane, therefore, reacts with Lewis bases such as dimethyl sulfide to form an acid-base complex. This we call an adduct, in which all of the atoms have a filled shell of valence electrons. Here is a summary slide that includes some organic and inorganic examples of Lewis bases and Lewis acids. The overarching theme of this slide is that all of the Lewis bases, no matter how they're defined, are acting as electron pair donors. The source of these electrons is the highest occupied molecular orbital of the species, whereas all of the Lewis acids, with their numerous examples, all have a LUMO, that's the lowest unoccupied molecular orbital. In an earlier video, you may recall that I talked about the overlap of the HOMO and LUMO of two reacting species to form a new covalent bond. This applies directly to this slide. Earlier in this video, I referred to water as being a Bronsted base and also as a Lewis base. However, water is an interesting molecule. Why? Let's take a look. Here we can see water behaving in two different ways. In the first scenario, shown at the top as a recap, water behaves as a Bronsted base and also as a Lewis base. The Lewis model is reflected by the movement of the electron pair using the curved arrow formalism. The tail of the arrow starts from an electron source, these are the lone pairs, and are nucleophilic, which are seeking a nucleus. The arrow head looks for an electrophilic, i.e. an electron-loving atom. In this case, the hydrogen atom of the hydrochloric acid. This is an electrophilic site. Of course, the hydrogen can't have two bonds, and so the bond between the hydrogen and chlorine is broken, with the more electronegative chlorine picking up the electron pair. This forms the conjugate acid-base pairs. Water behaves as a Bronsted base as it accepts a proton from hydrochloric acid. Hydrochloric acid has behaved as a Bronsted acid because it has donated its proton. Both models are reflected by this reaction. In the second scenario, something else happens. This time, ammonia is a Bronsted base and a Lewis base, nucleophile, as the arrow starts from the nitrogen's lone pairs and seeks a nucleus. The arrow head at the electrophilic hydrogen atom of water. The bond between the hydrogen and oxygen breaks as an additional nitrogen and hydrogen bond is formed. This way, all of the atoms now have a full valence shell, and in this case, water has acted as an acid. In the Bronsted-Lowry model, we would say that ammonia accepted a proton, whilst water donated its proton. Accordingly, we have another conjugate acid-base pair. Can you identify the conjugate acid and base for the second reaction? Pause the video and resume to check your answer. OH- is the conjugate base of water, and the ammonium ion is the conjugate acid of ammonia. What's interesting here is that water has behaved in one case as the base and in the other case as the acid. So we refer to compounds that are capable of doing this as being amphoteric. Now, seeing as you're studying biology, we should take a look at this phenomena again using a biomolecule. Here's that biomolecule that I was talking about. This is the amino acid glycine, and in glycine there are two groups, an acid group and a base group. Now, when placed in acidic media, the amine group will accept protons because it's basic and will form ammonium ions. They become protonated. However, when placed in basic media, the acidic protons of the carboxylic acid react with the hydroxide ions and they become deprotonated. Amino acids are described as being amphoteric compounds as they can either react with a base or an acid.
This concludes part A.
In this video we're going to look at the strengths of organic acids and bases. Let's consider the inorganic reaction first though. This is the acid-base reaction between hydroiodic acid and water. Hydroiodic acid protonates water to give hydronium ions and iodide anions as the conjugate acid-base pair. The reverse reaction can also occur. The iodide anions react with the hydronium ions to give hydroiodic acid. We say that the reaction is in equilibrium, but we use an acidity constant Ka. So we write this in the same way that we would write an equilibrium expression, and the concentration of water is so fast that it doesn't change and we ignore this. Now Ka is used to represent the equilibrium constant for the dissociation of the acid into its conjugate base and conjugate acid, and we can measure this equilibrium constant of a reaction, and it then tells us the relative acidity of hydroiodic acid. Now for this acid, it turns out that the equilibrium constant is about 10 billion. That means for every molecule of hydroiodic acid there are 10 billion iodide anions. In other words, this acid donates its proton very readily to become its conjugate base, which is typical of a strong acid. The smaller equilibrium arrow represents this. However, in reality, this reaction lies heavily to the right, and we can consider the acid as being fully dissociated in solution. So usually if you see these reactions, they would be represented by a full arrowhead. Let's take a look at some more reversible acid-base reactions. For the purposes of comparison, I've included the strong acid hydroiodic acid again. Here we said that 10 billion molecules are in the ionized form. But let's take a look at methanol, which is a considerably weaker acid. The acid dissociation constant of this alcohol is ridiculously small. Look at the order of magnitude here. That's 10 to the power of negative 16. Okay, let's rephrase this. For every 3,125,000 billion molecules of methanol, only one is in the ionized form. It's pretty safe to say this is a weak acid, so it won't readily dissociate. What about if we consider methane next? This really isn't an acid at all, but it drives home the point about the acidity dissociation constant and the comparative size of the equilibrium arrows. For every 10 to the power of 50 methane molecules, only one of them has the potential to be ionized. I include the number of zeros on the slide just to drive this point home. These numbers are either ridiculously large, I mean look at the Ka for hydroiodic acid, or ridiculously small. Look at methane. Our brains are just not geared for dealing with numbers on this scale. Therefore we adopt a different scale that contains the same information, just in a friendlier way. A more convenient measure of acidity is to use the negative logarithm of the acidity dissociation constant Ka. This is defined as pKa. When we now compare the extremely strong acid hydroiodic acid with the ridiculously weak acid methane, we have -10 for hydroiodic acid and 50 for methane. These numbers are far easier to cope with and also allow us to cover 60 orders of magnitude. Each pKa unit's increase or decrease reflects an order of 10. Let's take a look at some examples using an abbreviated pKa table. You have been provided with a full scale table on Canvas. Please use that one for any assessment, as the following slide is just for the purposes of demonstration. To make sense of what we've been discussing, let's get acquainted with this abbreviated version of a pKa table. The one you have from Canvas is going to be your friend on this course. In this one document, you'll get information on the scope and magnitude of a wide range of chemical behaviour, the strongest of the strong acids and the weakest of the weak acids. And since the stronger the acid, the weaker the conjugate base, these values also provide useful information about basicity. Let's consider a couple of examples of how to use this table. When comparing acidities of molecules, the lower the pKa value, the more acidic the species, and vice versa. Then the weaker its conjugate base, and vice versa. Even if we have a molecule that is a different chain length to our representative compound, the pKa table will still provide an estimate of your representative molecule. One question for you: how much more acidic are alcohols than ketones based on this table? Pause the video and resume when you have an answer. Alcohols, according to this abbreviated table, have a pKa of about 16, while ketones have a pKa of about 19. Now this is a difference of 3 pKa units. Remember, each pKa unit represents a power or order of 10, hence 10 to the power of 3 is 1000. Therefore, alcohols are 1000 times more acidic than ketones. Next question. Is the conjugate base of phenol a weaker or stronger base than the conjugate base of the alcohol? Pause the video and resume when you have an answer. There is an inverse relationship between the strength of the acid and the strength of the conjugate base. Phenol has a lower pKa than the alcohol. This makes its conjugate base, phenoxide, a weaker base than the alcohol beneath it. This would be an alkoxide. Finally, another way of using this table is in the discussion of protonation, that's adding a proton to a substance, or deprotonation, removing a proton from a substance. Provided there is a big enough difference in pKa unit, conjugate bases towards the bottom of the table will deprotonate the substances above them, and acids towards the top of the table will be able to protonate functional groups with acceptor atoms beneath them. Remember, this is not the complete picture of the substances in the pKa table. Let's take a look at some of these acid-base reactions now to see how difference in pKa provides a good predictor of how favourable an acid-base reaction is. Hydrochloric acid and sodium hydroxide react to give water and sodium chloride. But how favourable is this reaction? The pKa of hydrochloric acid is -8, so it's a very strong acid. The conjugate acid of sodium hydroxide is water, which has a pKa of about 16. That's a difference of about 24 pKa units, and since each pKa unit represents one order of magnitude, this reaction is favourable with an equilibrium constant of about 10 to the power of 24. For all intents and purposes, a reaction with an equilibrium constant of this magnitude means that the reaction will lie predominantly to the right, in other words, be fully dissociated. However, as a rule of thumb, reactions with a difference in pKa of about 10 units are irreversible. That is to say that hydrochloric acid and sodium hydroxide become completely consumed when they react together, giving only water and sodium chloride. One important takeaway from this slide is that the acid-base reactions always favour the formation of the weaker acid and the weaker base. Therefore, calculating the difference in pKa values is a useful predictor of equilibrium in an acid-base reaction. Okay, so what about a reversible example? Bases deprotonate, remove a proton from an acid. Being able to predict the strength of the base's ability to carry out such acid-base reactions is incredibly useful for later chapters. The pKa table will help you when you're considering the outcome of any acid-base reaction that leads to a conjugate acid-base pair. In this case, we're attempting to use sodium hydroxide to deprotonate methanol. Neither side of the acid-base reaction is strongly favoured. Here we're dealing with a very small difference in pKa, only about 0.2 pKa units. This is an entirely reversible equilibrium and so would not be synthetically useful. We may be able to figure out what's going on based off the previous slides, which is great, but for completeness, let's go through this example. First things first, you have to identify the acid and the conjugate acid to make use of their pKa values. In this case, acetic acid is, well, yes, an acid. But also look at the curly arrows. The proton is being donated to water. Also, the hydrogen atom is behaving as an electrophilic atom and is accepting an electron pair from water. Therefore, the suggestion is that the conjugate acid is the hydronium ion. The hydronium ion would have to donate the proton back to the acetate anion to reform acetic acid. Note I used the word suggestion though. Already if you're looking at the equilibrium arrows, you might be thinking it's the other way round. You're right, but humour me. When you look at the pKa values of the acid and air quotes conjugated acid, it's now possible to tell that the hydronium ion is the stronger acid. It has a pKa of negative 1.74, and that the acetic acid is the weaker acid. It turns out that water is not a strong enough base to deprotonate acetic acid. Therefore, the acetic acid is the conjugate acid, and the hydronium ion is the stronger acid. However, the acetate anion is a strong enough base to deprotonate the hydronium ion. Also, looking at the difference in magnitude between these two pKa values, this is about 6.5, and so means that the hydronium ion is 10 to the power of 6.5 times more acidic than the acetic acid. And as in the previous cases, the equilibrium moves from the side that contains the stronger acid to the side that contains the weaker acid.
This concludes Part B.